I want to start today with a number. And it is a number that is honestly hard to wrap your head around. 200 million. That is the conservative, the lower end estimate of how many human lives have been saved by a single discovery.
It's a statistic that just stops you in your tracks.
Yeah.
200 million. I mean, to put that in perspective for you, that's roughly the entire population of Brazil.
Right.
Or, the combined populations of the UK, France, and Germany. All of those people alive because of one specific moment in a laboratory.
And usually when we think of discoveries that shift the trajectory of the human species like that, we picture something like, a Manhattan Project scenario.
Sure. A gleaming high-tech bunker, a team of serious strategists, supercomputers, infinite funding, the whole thing.
The big science model. Exactly. Everything calculated, everything sterile. But this was not that. This story involves a messy, chaotic lab in London, a bit of stray mold, a window, or maybe it was a door. We're going to have to debate that. And a scientist who, instead of shouting, "Eureka!" just looked at a petri dish and muttered, "That's funny."
That's funny. I mean, it's amazing. 2 words that ushered in the entire antibiotic age.
Today, we are doing a deep dive into the life of Sir Alexander Fleming. And I mean the entire life, because I think people know the mold story, but they sort of treat it like a lightning strike.
Right, like he was just standing there and magic happened, a complete fluke.
Yeah. But when you look at the biographies, the scientific journals, the letters, the prequel and the sequel to that moment are just as wild. This isn't just about, a pill you take for a sore throat. This is the story of how humanity finally stopped dying from simple scratches.
It really is the dividing line in medical history, isn't it? There is the pre-antibiotic era where a rose thorn scratch could actually kill you, and frequently did. And then there's the post-antibiotic era. Fleming is the man standing right on that line. But what I'm really keen to unpack today is that Fleming wasn't just some guy who got lucky once. There's a whole lifetime of preparation that goes unnoticed. It's like Louis Pasteur said, chance favors only the prepared mind. We need to see how Fleming's mind got so prepared.
So we've got a massive stack of sources here. We're going to look at his rural Scottish roots, his miserable time as a shipping clerk, which actually explains a lot about his work ethic, the horrors of World War I that completely radicalized his view on medicine, and of course, the silent decade.
Oh, the silent decade. It's unbelievable. The most important drug in history just sat on a shelf. And we absolutely have to give credit to the team that turned that mold into actual medicine, because that is where the story gets really complicated. The Fleming myth often overshadows the brilliant chemists and engineers who did the heavy lifting.
We will get to all of that, but let's rewind all the way back, before the knighthood, before the Nobel Prize, before any of the petri dishes. Let's go to the Scottish moors.
Okay.
Alexander Fleming was born August 6, 1881 at Lochfield Farm in Ayrshire, Scotland.
A very humble beginning, a quintessential 19th century rural upbringing. He was the third of 4 children from his father's second marriage, but you had 8 children in total in that household.
So a crowded house.
Oh, a crowded, lively, probably very chaotic house.
And tragedy struck pretty early for him, didn't it? His dad, Hugh Fleming, died when Alexander was only 7 years old.
That's right. His mother, Grace, was left to manage the entire farm. And you just have to imagine the resilience that required back then. But looking at the sources, despite that hardship, there's this recurring theme about his childhood. He spent it roaming the moors.
Right, he wasn't inside reading books all day.
No, not at all. He was out there hunting, fishing, exploring the valleys. He was a creature of the outdoors.
It sounds kind of idyllic in a way, but the biographers all mention that this specific lifestyle honed his powers of observation.
And that is so crucial. It's not just a poetic detail. Think about what it takes to be a good hunter or a good fisherman out in the Scottish wild. You have to notice the tiniest changes in the environment.
A bent twig, a ripple in the water.
Exactly. A change in the wind. You have to see things that other people just walk right past. Fleming carried that exact skill, that habit of mind, straight into the laboratory. He wasn't the kind of scientist who just looked for what he expected to find. He noticed what was actually there, even if it didn't belong.
So he's this farm kid with eagle eyes, but he doesn't stay on the farm. At age 13, he makes the move to London to live with his older brother, Thomas.
Which must have been a massive culture shock, from the open, clean air of the moors to the smog and congestion of Victorian London.
Yeah.
He goes to school at Regent Street Polytechnic. But here is a twist that really surprised me when I was digging into the timeline. He doesn't go straight into medicine.
No, and this is the part that makes you realize how fragile history is. The man who saved the world spent 4 years working as a clerk for a shipping office.
A shipping clerk. And by all accounts, he absolutely hated it.
I mean, I can imagine. Just pushing paper all day.
It was unfulfilling, repetitive, administrative drudgery. He was just another cog in the imperial trade machine, dealing with invoices and manifests. Total dead end.
It's so weird to think about. If he had been just a little bit more complacent or, if he'd gotten a decent promotion or something, he might have just stayed there.
He would have been Alexander Fleming, the senior logistics manager.
And we might all still be dying of sepsis from a paper cut.
It's a terrifying thought. But luck or fate or maybe just family pressure intervened. At age 20, he received a small inheritance from an uncle, John Fleming. It wasn't a fortune, but it was enough to buy his freedom.
And his brother stepped in.
Right. His brother Thomas, who was already an oculist, an eye doctor, basically sat him down and said, look, you're smart, you have this money, get out of that office and go study medicine.
So he takes the advice. He sits for the entrance exam for medical school. And of course, because he's Alexander Fleming, he doesn't just pass.
He places first in the entire United Kingdom.
First. That's the level of intellect we're dealing with here. So he has his pick of any medical school in London. He could go anywhere he wants. And this is where we see just how random life can be.
Right.
Why did he choose St. Mary's Hospital Medical School?
Was it their prestigious faculty?
No, they're cutting-edge labs.
No, he had played water polo against them.
You are kidding me.
No, I'm serious.
Yeah.
He liked their water polo team. He thought they were a good bunch of guys. That is literally how he chose the institution where penicillin would eventually be discovered.
That is hilarious. But there's an even deeper, more specific reason he stayed at St. Mary's, and it has nothing to do with science and everything to do with another sport.
Ah, yes. The Rifle Club pivot. This is one of my absolute favorite anecdotes in the history of science.
Okay, let's unpack this, because it sounds like a plot point from a movie that critics would say is just too unrealistic to believe.
So it's 1906. Fleming is qualified as a surgeon. He's got his MBBS with distinction. He's brilliant. The standard path for a man with his talents is to leave the medical school, go out into the world, join a practice on Harley Street, and become a wealthy, successful surgeon.
Right. Big money, prestige. That's the dream for any young doctor.
But Fleming had joined the St. Mary's Rifle Club. And remember those powers of observation he honed on the moors? It turns out they translate perfectly to marksmanship.
He was a good shot?
He was a recognized marksman. He was essentially the star player on the team. The captain of the Rifle Club was a man who took winning very, very seriously. And he knew that if Fleming left to become a surgeon, he'd have to leave the club and the team would lose their best shot.
So the captain intervenes?
He pulls strings. He practically begs the hospital administration. He convinces Fleming to join the research department, the inoculation department, right there at St. Mary's. Not because he saw Fleming's potential as a great researcher, but to keep him in the building for the shooting team.
That is absolutely wild. If he had been a bad shot, he would have been an anonymous, wealthy surgeon. And penicillin just sits undiscovered in some mold for another 100 years.
Exactly. We owe the entire antibiotic age to Fleming's steady hand with a rifle. And the research department he joined, well, it was run by a titan of the field, Sir Omroth Wright.
Let's talk about Wright. Because to understand Fleming, you have to understand the guy he was working for. Wright wasn't just a boss. He was a mentor with a very, very specific dogma.
Oh, absolutely. Wright was a dominant personality, a pioneer in immunology and vaccine therapy. His whole philosophy was about stimulating the immune system.
Right.
He believed in vaccines. He believed the body had all the tools it needed to fight infection. You just had to wake them up. He was actually quite skeptical of chemical drugs. His motto was essentially, the doctor's job is to help the body help itself.
So Fleming is in this environment, absorbing this philosophy, the body knows best.
He's absorbing the rigorous thinking, yes. But Fleming also had a side gig. He had a private practice where he was treating syphilis. And at the time, the treatment for syphilis was a drug called Selverson.
Paul Ehrlich's magic bullet.
Precisely. It was a chemical that you injected, and it killed the bacteria without killing the patient. A true magic bullet.
So Fleming is seeing these two different worlds collide. In the lab, his mentor Wright is saying use the immune system, stimulate it. But in his private practice, he's seeing a chemical, a drug, actually work to kill bacteria directly.
Exactly. It planted a seed in his mind. He realized that chemical agents could work. But he didn't really have time to develop that thought because history intervened again in a big way. World War I breaks out.
And this is the crucible, isn't it? This is where the theorist becomes the practitioner in the worst possible way.
Yes. Fleming serves as a captain in the Royal Army Medical Corps. He's stationed at a casualty clearing station in Boulogne, France. And we have to be really clear about what he saw there. It wasn't just the trauma of the bullets and the shells, it was what happened afterwards.
The infections.
The infections. The battlefields of WWI were literally farm fields. They had been fertilized with manure for centuries. They were unbelievably dirty. When a soldier got hit, shrapnel would drive mud, bits of fabric, and manure deep into the muscle tissue.
A breeding ground for bacteria.
The perfect breeding ground. Sepsis, gas gangrene, tetanus men were dying by the thousands, not from the initial injury, but from the raging infection that followed days later in the hospital.
And the standard practice at the time was to just use antiseptics, carbolic acid, things like that. Surgeons were just pouring it directly into the wounds, right?
Right. And on the surface, it makes intuitive sense. If you have a dirty floor, you use strong bleach. If you have a dirty wound, you use strong antiseptic. But Fleming, with those incredible powers of observation, he realized something horrific was happening.
What was that?
The antiseptics were killing the soldiers faster than the bacteria were.
Wow. That's a heavy statement. How is that even possible?
Well, Fleming devised this brilliant experiment to prove it. He was a skilled glassblower, another one of his many hobbies. He created an artificial wound model using glass.
What do you mean?
He blew glass into a shape that mimicked a jagged shrapnel wound, with all these little nooks and crannies and deep pockets.
So he could see what was happening inside.
He could see everything. You'd fill it with infected serum and then pour in the antiseptic. And he showed that when you do that, the antiseptic only cleans the surface. It can't penetrate into those deep, jagged recesses where the anaerobic bacteria are hiding and multiplying.
Okay, so it wasn't reaching the real problem.
It wasn't. But it gets so much worse.
Because of the white blood cells.
Yes. The antiseptics were absolutely devastating to the body's leukocytes, the white blood cells. Those are your body's natural defenders, your frontline soldiers. So picture this. You pour carbolic acid into a deep wound. You fail to kill the deep bacteria because the acid can't reach them.
Right.
But the acid does reach and kill all the white blood cells that were gathering to fight the infection. You're essentially disarming the patient and leaving the bacteria untouched in their bunker.
That is just tragic. You're literally clearing the way for the enemy to take over.
It was a medical disaster. Fleming published these findings. He practically screamed it from the rooftops. Almaroff Wright supported him. But the military doctors, the old guard, they completely ignored him. They kept pouring carbolic acid into wounds for the entire duration of the war.
That must have been so infuriating to know, you're right, to have the data and to just stand by and watch men die because of protocol.
It was traumatizing. The sources make it clear. Fleming returned from the war absolutely obsessed. He was determined to find a natural antibacterial agent, something that would kill the bugs, but, and this was the crucial part for him, would not harm human cells. That became his holy grail.
Non-toxic to us, lethal to them.
That was the mission.
So he's back at St. Mary's. It's the early 1920s. He's on the hunt for this magic substance. And before we get to the main event, there's a sort of prequel discovery, isn't there? The snot discovery.
The snot discovery, yes.
Yes.
1921. This is often called the rehearsal for penicillin. It's a fantastic story.
So set the scene for us.
Well, it's pretty simple. Fleming has a severe cold. He's, dripping. And being Alexander Fleming, he decides to conduct an experiment on himself.
Yeah.
He takes a drop of his own nasal mucus and lets it fall onto a culture plate that's teeming with bacteria.
Gross. But very scientific.
Very scientific. He incubates the plate. And when he checks it later, he sees something amazing. Everywhere the mucus had touched, the bacteria dissolved. They were just gone, wiped clean.
So she discovered lysozyme.
Exactly. Lysozyme. It's an enzyme. It's in our tears. It's in our saliva, our mucus, even in egg whites. It's part of our body's natural antiseptic defense system, our innate immunity.
But it wasn't the cure-all he was looking for, was it?
No, and that was the big disappointment. Lysozyme is great at killing harmless airborne bacteria, but against the really heavy hitters, the pathogens like Staphylococcus or Streptococcus that were killing people in the hospitals, it didn't really do much.
Why not?
The cell walls of those dangerous bacteria were just too thick and complex. Lysozyme couldn't break them down, but, and this is the key thing, it proved the concept. It proved that there were natural substances out there that could dissolve bacteria without hurting human cells. It kept his mind prepared and looking for the next thing.
Speaking of prepared minds and unusual hobbies, we have to talk about Fleming the artist, because this is my favorite weird fact about him. He was a member of the Chelsea Arts Club.
He was. an amateur watercolorist. But he took that artistic impulse into the lab. He created what we now call microbial art.
He literally painted with germs.
He painted with germs. He would take a Petri dish as his canvas, and instead of paint, he would use different bacteria that had different natural colors. Serratia marcescens for red, Chromobacterium violaceum for purple, Micrococcus luteus for yellow.
And he'd make actual pictures.
Oh, elaborate pictures. There are photos of them. Ballerinas, the Union Jack flag, stick figures fighting, even a mother feeding her child.
Wow.
And the technical skill involved was insane. You see, these different bacteria grow at different speeds. So if you want the red and the yellow parts of your painting to appear at the same time, you have to paint the slow-growing ones first, wait a day or so, then paint the fast-growing ones.
That is just an incredible level of dedication. It shows he really knew the life cycles of these organisms intimately.
It showed his complete mastery of the Petri dish. He knew exactly how these things grew, how they behaved. There's a funny story that he made a Union Jack out of bacteria for a visit to the lab by Queen Mary.
Did she like it? Was she impressed?
Reportedly, she rushed past it, completely unimpressed. I guess royal appreciation for bacterial art was a bit low in the 1920s.
Her loss. But you can see how all these things are converging. The observation from the Moors, the WWI obsession, the lysozyme concept, the mastery of the Petri dish. It all leads us to the big moment.
The aha moment.
Yep. The date is September 1928. Fleming is back from a holiday.
Right. So at the time, Fleming is researching Staphylococcus. He's looking for variations in the bacteria. Now, we have to address his lab habits.
He was messy.
He was notoriously messy. He didn't throw his culture plates away immediately. He would stack them in trays of Lysol, but he let them pile up so high that the top ones were sitting high and dry, completely above the disinfectant.
So he goes on holiday with his family to Suffolk. He leaves a stack of these undisinfected plates sitting on the bench.
And while he's gone, something happens. A spore of mold lands on one of the plates. Now, the traditional story, the one you'll see in old textbooks and movies, is that the spore floated in through an open window from Parade Street.
The romantic notion of nature just drifting in on the breeze to save humanity.
It's a lovely poetic idea, but it's probably wrong. Bacteriologists in London rarely open their windows because, well, you don't want contamination from all the soot and dust of a busy street.
So where did it come from?
The expert consensus now is that the spore likely came from the mycology lab, the mold lab, which was on the floor directly downstairs, run by a man named C.J. Latouche.
So it just drifted up the stairs?
It likely drifted up the stairwell, or maybe Fleming or an assistant carried it in on their clothes after visiting Latouche's lab. Latouche was actively working with various molds to research allergies. It's a much more plausible source than the magic window.
So Fleming comes back from his holiday. It's September 3rd-ish. He's chatting with a colleague, a guy named D.M. Price, and he picks up one of these discarded plates from the pile.
He's about to toss it into the Lysol. But he looks at it. He sees a blob of greenish-blue mold. But that's not the interesting part. Mold grows on bread, cheese, whatever. It's common.
The interesting part was what was happening around the mold.
Exactly. He saw what he called the ghostly appearance of the bacteria, the ring of death.
Explain that. What did it look like?
Around the mold, the colonies of Staphylococcus, which usually look like opaque yellowish dots, had become transparent. They were dissolving. They were undergoing lysis. The bacteria were literally exploding.
Can we just pause on that for a second? Exploding? What is actually happening biologically there to the bacteria?
It's terrifyingly effective if you're a bacterium. Penicillin works by attacking the enzymes that build and repair the bacterial cell wall. Think of the cell wall as a kind of corset or a suit of armor that holds the bacterium together under pressure.
Okay.
The bacterium keeps trying to grow and expand, but the penicillin prevents it from building new armor. So the internal pressure builds up and up until pop. The cell wall ruptures, and the bacterium essentially spills its guts out. It tricks the bacteria into committing suicide by growth.
And the reason it's safe for us is what exactly?
Human cells don't have cell walls. We have cell membranes, which are a completely different structure. So penicillin is like a sniper shot that hits the enemy's specific infrastructure, but passes right through our own city without doing any damage.
That is just incredible. And when Fleming saw this ring of death, he didn't shout Eureka.
No, he just murmured, "That's funny."
That's funny.
That is the absolute essence of scientific discovery, isn't it?
Yeah.
It's not about finding what you're looking for. It's about noticing when something weird and unexpected happens and having the curiosity to ask why.
So he immediately identifies the mold as a species of Penicillium.
It was eventually identified as Penicillium notatum, and he names the juice it's exuding penicillin.
And he runs a battery of tests on it?
He does. And the results must have been mind-blowing for him. It kills the bad guys. Scarlet fever, pneumonia, gonorrhea, meningitis, diphtheria. These were the absolute killers of the age.
Does it kill everything?
No, which was also interesting. It didn't touch typhoid, which is a gram-negative bacterium. Gram-negative bacteria have a different kind of outer shell that the penicillin couldn't penetrate easily. But against the gram-positive bacteria, it was a nuke. And crucially, he injected it into a rabbit. He put it on human white blood cells in a dish.
It didn't hurt them?
Nothing. It was completely non-toxic. It was the magic bullet he had been dreaming of ever since the trenches of France.
So this is it. 1929, he publishes the paper. He has the cure for pneumonia, for gonorrhea, for meningitis. The world must have gone absolutely crazy, ticker tape parades.
You would absolutely think so. But this brings us to one of the most baffling parts of this story, the silent decade. He publishes his findings in the British Journal of Experimental Pathology. And the reaction from the scientific community is silence.
Crickets. How do you ignore the cure for everything?
It's baffled historians for years. But you have to try to put yourself in the mindset of a 1929 doctor. They had been burned by so-called miracle cures before. Vaccine therapy, Almroth Wright's philosophy, that was still the dogma.
Right.
The idea that a mold juice, something that's associated with decay and rot, could outperform the body's own sophisticated immune system seemed almost backward, almost like folk medicine. When he presented his findings to the prestigious Medical Research Club, literally no one asked a single question afterward.
They just saw it as a lab curiosity.
A cool lab trick, not a revolution in medicine. But there was a huge technical problem, too, which was the real barrier.
The chemistry.
The chemistry. Fleming was a brilliant bacteriologist, but he was not a chemist. Penicillin, as it comes out of the mold, is incredibly unstable. It degrades in a matter of hours at room temperature. If you try to heat it to concentrate it, it vanishes. If you change the pH too much, it breaks down.
So he couldn't turn the mold juice into a stable pill or an injection that you could actually use on a person.
Exactly. He couldn't purify it. He couldn't concentrate it in any meaningful way. So for 10 years, from 1929 to about 1939, penicillin was mostly used as a clever lab tool.
Mm-hmm.
Fleming would use it to kill off unwanted gram-positive bacteria in mixed cultures so he could isolate the penicillin-insensitive ones he wanted to study.
That is just heartbreaking. It's like finding a diamond and using it as a doorstop.
It is. But Fleming did one absolutely crucial thing during this decade. He kept the mold alive. He was like a zookeeper for this one specific strain of penicillium. For 12 years, he grew it, he subcultured it, and he sent samples of it to anyone who asked, free of charge. He kept the strain extant for the future.
Did he try to use it on people at all during this time? There must have been some attempts.
A few, yes. There was a colleague, Dr. Stuart Craddock, who had a nasal infection. They tried it, but it failed. Most likely because the bacteria was resistant or the drug just wasn't concentrated enough to work. But there was a success. A former student of his named Cecil George Payne, working in Sheffield, used the crude mold juice in 1930 to cure babies with eye infections, ophthalmia neonatorum. It worked.
But that didn't make headlines.
Payne never published it widely. He was a quiet, practical physician, not a researcher looking for fame. And then there's a great story about a man named Keith Bernard Rogers. He had conjunctivitis and was about to drop out of a rifle shooting competition.
The rifle theme returns.
It always comes back. Fleming gave him some penicillin ointment, he cleared up his eye infection, and Rogers went on to win the competition.
So it worked. The proof was there in these small cases, but it just wasn't scalable.
Not yet. It needed a completely different set of skills to unlock its potential. It needed a team of chemists and industrialists.
Enter the Oxford team. This is where the story shifts gears from a solo discovery to what is essentially an industrial revolution in medicine. We need to introduce these characters because they're just as important as Fleming.
Absolutely. It's now 1939. War is looming over Europe again. At Oxford University, you have a dream team forming. First, you have Howard Florey, an Australian pathologist. Brilliant, driven, incredibly ambitious, and frankly, a bit abrasive. He was the manager, the force of nature who pushed the project through.
And then the chemist, the guy who could solve the problem that stumped Fleming.
Ernst Chain, a Jewish refugee from Nazi Germany. He had fled to Britain with only 10 pounds in his pocket. He was a genius biochemist, but also incredibly intense, emotional, and brilliant. So Chain is digging through old journals, what he called the graveyard of scientific literature, looking for a new project.
And he finds Fleming's 1929 paper.
He finds the paper on penicillin, And he looks at it from a chemical perspective. He thinks, this enzyme potential is interesting. I think I can stabilize this molecule.
So they decide to work on it. Does Fleming even know this is happening?
Not at first. But when Fleming hears through the grapevine that the heavyweights at Oxford are working on his old mold, he catches a train and goes to visit them. He just walks into the lab. And Jane's reaction is legendary.
What did he say?
He reportedly said, "Good God, I thought he was dead."
That's amazing. Fleming had been so quiet on the subject for a decade, they just assumed he was history.
But Fleming was happy to help. He handed over his original cultures, and the Oxford team went to work. And this is where the messy lab bench at St. Mary's turns into what you could only call a primitive factory.
They hired a team they called the Penicillin girls, right?
Yes, a group of young women were hired to manage the day-to-day growing of the mold. But the conditions were incredibly primitive. They couldn't get proper funding. It was wartime, all resources were going to the military, so they had to improvise. And we have to talk about Norman Heatley.
The unsung hero of the story.
The absolute unsung hero. Heatley was the MacGyver of the group, a brilliant biochemist who could build anything out of anything. He built an extraction apparatus using old bookshelves, doorbell wire, and milk churns. But the biggest problem was just growing enough mold. It needs surface area to grow. It forms a mat on top of the liquid.
So they needed thousands of shallow pans.
Thousands. But they couldn't buy thousands of glass flasks. So Heatley designed custom ceramic vessels. They went to a pottery firm in Stoke-on-Trent and ordered hundreds of them. And the thing is, they look exactly like hospital bedpans.
So the world's first antibiotic factory was a room full of bedpans in Oxford.
That is correct. They stacked them in the animal operating theater. The whole place smelled terrible, but it worked. Heatley figured out the genius method of back extraction, changing the acidity to pull the penicillin out of the liquid and into a solvent, and then back into water, concentrating it each time.
So they finally have enough of this concentrated brown powder, that's what it looked like, right? To test on mice. This is May 1940.
Probably the most important mouse experiment in history. They took 8 mice. All of them were injected with a lethal dose of streptococci. 4 of the mice get the brown powder, the penicillin. 4 of them get nothing.
And the result?
Jane and Heatley stayed up all night watching them. The next morning, the 4 control mice are dead. Rigor mortis has set in. The 4 treated mice are alive, hopping around, eating their breakfast as if nothing happened. Jane called it a miracle.
But mice aren't men. They need a human patient. And this story, this is the one that really breaks your heart.
Albert Alexander, February 1941. He was a 43-year-old policeman. And the story goes, he scratched the side of his mouth on a rose bush in his garden.
A simple rose bush scratch.
That's all it took back then. The scratch got infected with both staphylococcus and streptococcus. It spread. His face was covered in horrific abscesses. He had already lost an eye to the infection. He was dying of sepsis.
They give him the penicillin.
And it works like magic. The effect was immediate. Within 24 hours, his fever dropped. The abscesses began to heal. He started to sit up in bed. He was eating again. It was undeniably a cure. But
they didn't have enough.
It was still so incredibly hard to make in those bedpans. They simply ran out. They were desperate. So they started collecting his urine.
His urine? Why?
Because the human body excretes penicillin almost completely unchanged. So they would collect his urine, bicycle it back to the lab, have the chemists extract the precious penicillin out of it, purify it, and then run it back to the hospital to re-inject into him. They were literally recycling the drug through their patient.
That just shows the sheer desperation of the situation.
Yeah, it does. But eventually, even that wasn't enough. The supply ran out completely. The infection came roaring back, and Albert Alexander died.
That is just devastating. To prove it works, to see the man recover, and then to lose him simply because of supply.
It was a brutal lesson. But it proved the drug worked in humans. And it proved they couldn't make this in bathtubs and bedpans in a university. They needed massive industrial scale. But British industry was being bombed by the Luftwaffe. They couldn't build new factories.
So they look west. To America.
Florey and Heatley fly to the U.S. in a blacked-out plane, carrying a vial of the mold in their pockets. They go to Peoria, Illinois, to a USDA agricultural research lab, and this is where the second miracle of penicillin happens.
Deep tank fermentation.
Right. The Americans were experts in large-scale fermentation for things like citric acid. They figured out that if you use corn-steeped liquor, which is basically a sludge byproduct from making cornstarch that they were just dumping in the river, the mold absolutely loves it. It grows like crazy.
And they still needed a better strain of the mold, right? Fleming's original was a bit of a wimp when it came to mass production.
They started a global search. They had Army pilots scooping up soil samples from airfields all over the world. But the winning strain It came from a market right there in Peoria.
You're kidding.
A lab assistant named Mary Hunt, forever known as Moldy Mary, was tasked with finding moldy food. She brought in a moldy cantaloupe she found at a local grocery store.
A cantaloupe from a grocery store.
That mold on that cantaloupe, Penicillin chrysogenum, produced thousands of times more penicillin than Fleming's original strain. It was the key to mass production.
From a cantaloupe in Illinois to the beaches of Normandy.
Exactly. The American industrial machine kicked into high gear. Companies like Pfizer, Merck, Squibb, they developed the deep tank fermentation methods. By D-Day in 1944, they had produced enough penicillin to treat every single wounded Allied soldier. It was a massive strategic advantage. The Germans didn't have it.
It treated gangrene, it treated syphilis. I saw there were adverts in military papers saying things like, penicillin cures gonorrhea in 4 hours.
It completely changed the behavior of the entire army, and it changed the survival rate of the war. It was the first time in human history that infection didn't claim more lives on the battlefield than battle itself.
So now we're after the war. Fleming is a global superstar, the penicillin man.
He became the face of penicillin. The press absolutely loved him. He was modest, he was Scottish, he had that perfect accidental genius vibe. He was much more photogenic and interview-friendly than the, let's say, more abrasive Howard Florey.
But that created some tension, didn't it, with Florey and Chain and the rest of the Oxford team?
Oh, huge tension. Florey and Chain felt completely overshadowed. They had done the incredibly difficult heavy lifting of turning a lab curiosity into a life-saving drug. They figured out the chemistry, the extraction, the clinical trials, the industrial production. Yeah. But the public loves a lone hero story.
The Fleming myth.
The Fleming myth was born. Now, to Fleming's credit, he always acknowledged them publicly. He would say in speeches, "I discovered the mold, they made the drug." But the newspaper headlines were always just Fleming.
In 1945, they share the Nobel Prize in Medicine, Fleming, Florey, and Chain.
Correct, which was the right thing to do. But even then, the myths just kept swirling. We have to bust the big one. The Winston Churchill story.
Oh, I love this one. I've heard this so many times, but you're going to tell me it's completely fake.
It is a total fairy tale. The story goes that Fleming's father, a poor Scottish farmer, saved a young Winston Churchill from drowning in a bog. So, in gratitude, Lord Randolph Churchill paid for Alexander Fleming's entire education.
And then, years later,
Winston Churchill gets pneumonia, and who saves him but Alexander Fleming with his new wonder drug, penicillin? It's just the perfect circle of fate.
It's too good to be true.
Fleming himself called it a wondrous fable. The truth is, Churchill was sick with pneumonia in 1943 in Tunisia, but he was treated with sulfonamides, the sulfa drugs that came before penicillin, not penicillin. And Fleming's education was paid for by his uncle's inheritance, not a grateful lord.
Myth officially busted. Okay, let's talk a bit about his personal life in these later years. He seems like a very quiet, modest man.
Very taciturn, not a great conversationalist. His first wife, Sarah, was a nurse, and she died in 1949, which hit him very hard. But he did find love again. In 1953, he married Dr. Amalia Koutsuri-Varica. She was a Greek bacteriologist and a colleague at St. Mary's. It seems to have been a true partnership of minds.
Yeah, there's one more critical thing we have to cover before we wrap up. The money, or the lack of it. Fleming didn't patent penicillin.
He didn't. None of the British team did. They believed it should be a gift to humanity. They wanted it to be free for everyone. It was a very powerful ethical stance.
That is incredibly noble.
It is. But the American companies did patent the methods for mass production, the deep tank fermentation process, which meant that, ironically, Britain eventually had to pay royalties to American companies to manufacture the drug that was discovered in a London hospital.
Ouch. That must have stung Fleming and Florey.
Fleming was furious about it.
Yeah.
There's a quote where he said, "I found penicillin and have given it free for the benefit of humanity. Why should it become a profit-making monopoly?"
He had a really good point. But even more prophetic, even more important than his anger about patents, was his warning about the drug itself.
This gives me chills every single time I read it. In his Nobel lecture, this is 1945, right at the very dawn of the antibiotic era, Fleming explicitly warns about resistance.
He saw it coming that early.
He saw the whole thing. He explained the mechanism perfectly. He said, and I'm quoting here, "The ignorant man may easily underdose himself and, by exposing his microbes to non-lethal quantities of the drug, make them resistant."
That is exactly what is happening right now, today, with MRSA and superbugs.
Exactly. He knew that if you didn't kill the bacteria completely, if you just wounded them and let them recover, you were essentially training them. You were selecting for the strongest ones, the ones that could survive, and they would pass that resistance on. He predicted the entire superbug crisis of the 21st century back in 1945.
That is the mark of a true genius, not just discovering the thing, but understanding the profound consequences of it, for good and for ill.
Absolutely. It's astounding foresight.
So Fleming passes away in 1955.
Yes, a heart attack, March 11th. He was 73 years old.
And he's buried in St. Paul's Cathedral, which is a huge national honor. That's where heroes like Lord Nelson and the Duke of Wellington are.
It fits, doesn't it?
Yeah.
Nelson saved the country from Napoleon. Fleming saved the entire species from bacteria.
When you look back at his whole life, from the moors to the shipping office, the trenches, the messy lab, the Nobel Prize, what is the big takeaway for you?
It's that concept we started with, the prepared mind. Fleming didn't invent the mold. Nature did that. Thousands of people probably saw moldy bread or moldy culture plates before him and just threw them out. Fleming was the only one who stopped, looked, and had the lifetime of preparation to understand that the death of the bacteria in that dish meant life for us.
He had the curiosity to say, that's funny, instead of just, that's dirty.
He turned a contamination, an accident, into a revolution. He single-handedly changed medicine from the passive observation of death to the active cure of life.
And he did it all with a bit of snot, a bit of mold, a messy desk, and a very, very steady hand with a rifle.
The lesson is, don't clean your room, kids. You might save the world.
There you have it. The incredible, accidental, and utterly persistent life of Sir Alexander Fleming.
A fascinating journey.
I want to leave you all with one final thought. Fleming painted pictures with bacteria. He saw the patterns, maybe even the beauty, in the very microbes that kill us. Today, scientists are using bioluminescent bacteria-glowing germs to do things like track cancer cells in the body and study how genes express themselves. In a way, they are still painting with Fleming's palette. We're still living in the world that he first sketched out on that little glass dish.
That's a beautiful image to end on.
If you enjoyed this exploration of the man who accidentally saved the world, make sure to like and subscribe to History Titans. We've got more stories of the people who shaped our reality coming up, from the inventors to the revolutionaries, so hit that notification bell so you don't miss out. Thanks so much for listening.
Goodbye, everyone.